So hello everybody, thank you for coming back after the break. And it's now my pleasure to introduce Victoria Rajewicz. I for sure pronounced this also wrongly. Talking about biohybrid entities that are used for environmental monitoring. The stage is yours, Victoria.
All right, thank you, Thomas. Thank you for the introduction. I'll stop the video, save some of that bandwidth. That's okay. Okay. I hope you can all hear me clearly. Okay. Hello, everybody. Good afternoon, good morning. My name is Victoria Rajewicz, and I'm a biologist. Yeah, it's okay. Happens all the time. I'm a biologist and a PhD student at the University of Graz here in Austria. And today, I'll be talking about some of the pros and challenges that come with building and working with biohybrid entities for environmental monitoring, which is what we're working on within the framework of our project, Robocenosis. So first things first, why the need for aquatic environmental monitoring in the first place? So I'm sure we all know that the Earth is faced with an immense ecological crisis. The water quality is worsening every year. The freshwater supply is also diminishing every year. And it's all very complex. So it's not just one thing causing it. It's global warming, industrialism, et cetera, mostly anthropogenic factors. So scientists basically agree that the only way to slow down the process or stop the process is extensive, continuous environmental monitoring and sharing of the world's water data. So now that we know that aquatic monitoring is necessary, what's the best way to do it? Even today and in the past, it's mostly been dominated by sampling, which is a perfectly valid method to monitor the environment, but it's very time-consuming, very money-consuming. It involves a lot of materials and also labor. So there's a lot of people involved in taking those samples and analyzing them later on in the lab. So all this leads to a slower processing time and also fewer data points. So this makes continuous monitoring almost impossible or just very difficult. Hence the need for robots. Robots are basically the only way or the best way for continuous real-time monitoring. And this is exactly what we're working on with the project Robocenosis. Here is an artistic representation of how the end product hopefully would look like. This is so-called biohybrid. So what is a biohybrid? It has a lot of definitions, but here we define it as a structure that will connect biological entities and materials that will allow to extract useful data from those entities. So we'll be using a concept called life form in the loop, meaning we will be using life forms or those biological entities for monitoring the environment. So again, here is an artistic representation. It will be composed of two modules. So our first main objective is, of course, biomonitoring. So using those life forms as biosensors, so instead of classical sensors. These sensors, for example, plants, algae, animals, or even bacteria, they will be continuously, all the time, interacting with the environment. And thanks to electronics that will be in one of the modules, we can extract data on the water quality from those life forms. Our next main objective is the biodegradability. So again, two modules. One of them is non-biodegradable, that will contain all the electronics, all the cameras, perhaps some sensors. And this module will be retrieved after the project is completed. And the sort of other module, including the platforms and the connective tissues, also, of course, the life forms themselves, will be completely biodegradable and will remain in the environment after the project has been completed. And our third main objective or challenge is this biohybrid's autonomy. So we want this robot to not need any external batteries. And we will be using that, for that we will be using energy harvesting and low power and electronics that I will talk about in a second. So some key data about Robocenosis. We are a European Union funded project. We have a budget of 3 million euros and a runtime of 5 years, and we are now closely approaching our first year. Here at the University of Graz, we are the coordinators, but we have the pleasure of working with universities in Pisa and in Brussels and in Manchester. We'll be focusing on various Austrian lakes, and they're all protected areas, so it's really great that we have the allowance to work there. And I will talk about them in a bit. So with Robocenosis, we are trying to answer many questions, but if I had to pick the two sort of most important ones, they would be, can we use living organisms instead of classical sensors as surrogates for classical sensors? So of course, living organism won't give us a number as a classical sensor would. So for example, if we used an oxygen probe, this would give us a number of the level of the oxygen. So a life form won't do that, but that is exactly the point. We wanted to collect data on not just one environmental factor, but the overall state of the environment. And second question is, can we use alternative energy sources to power this robot under real-time conditions in long-term aquatic monitoring? And as the alternative power source, we will be using microbial fuel cells, or MFCs for short, that I will explain in a second. So here is the mission plan. Here the robot is deployed, so it's sinking to the bottom of the lake. We could provide it with some life forms, but we mostly want the life forms to grow on it. Later, the robot gets adapted to the environment. As I mentioned, things grow on it. This is called fouling. So usually fouling is an obstacle in any underwater constructions or robotics. But here we want it to foul, like we will use it to our advantage. Later on, during the process, the robot can decide to explore some other parts of the lake, or maybe emerge and submerge itself a couple times. During the whole process, it's of course measuring and continuously sending us data. Even more organisms crawl next to it, swim next to it, grow on top of it. And after, let's say, a couple months or a year, the non-biodegradable module with all the electronics and data will float upwards, ping us that the research is done, and we will go collect it. And the rest is going to remain in the environment. So here, the robot is long gone, and everything is sort of back to natural state. Here is a more sort of sophisticated version of how the biohybrid entity will possibly look like. Again, here is the core. So all the electronics, maybe cameras, maybe some sensors will be within this core. And this core will be attached to some sort of balloon, any floating device, or it float on its own. So after the experiment, this is the part that will float upwards. And the rest, all the platforms, the power supply, the any connecting mechanisms here, and of course the life forms themselves, everything will be remain in the environment. So some of the life forms that we would like to use, or we're already using, are mussels. Of course, biologists love mussels as bioindicators because they're quite easy to collect, to work with. They don't move around too much, and they're also really good bioindicators because of how they feed. They're filter feeders. So we are focusing on two species. One is swan mussel right here, and this is one of the largest freshwater mussels. And it's also very sensitive to low oxygen, but it's also very rare. So we're mostly focusing on the zebra mussel, which is right here. It's much smaller. It's about 1 centimeter, but there's plenty of it in the lakes that we're working. So it's really beneficial to use it, and it's also an invasive species. Some other stressors that the mussels are sensitive to are temperature change, starvation, predator presence, irritation, and many others possibly. Of course, some of these are not as useful to us as others. For example, irritation, it just means that something tapped on it or crawled on top of it and it got stressed. But we can rule these out later on by data analysis. So for example, if it's just a one-time occurrence, it's probably nothing to worry about. But if the stressor occurs multiple times over a long period of time, then this is probably a sign of a pollution. Here's something I called difficulty of perception. So how easy or how difficult it is to tell that the organism is stressed. So how difficult it is to read the stress behavior. So here in this case, the difficulty is low, meaning it's quite easy to tell, because in case of a mussel, there is only one movement, which is the closing and opening of the valve. So they're pretty sedentary otherwise. And that's why it's pretty easy to tell if they're closed for a prolonged period of time. This is a sign of stress. Next up, we have cladocerans, especially Daphnia magna and Daphnia pulex, mostly because they are very heavily researched. So we know a lot about them, and we know that they are good bioindicators. They're very sensitive to, again, low oxygen, to pesticides, any rapid changes in temperature and pH, and many other factors. But here, the difficulty of perception is medium. It becomes medium because we're not looking at just one movement, like closing and opening of the valve. We'll be looking at many different things. So I'll be focusing on, for example, swimming pattern. So their distribution, any erratic swimming. For example, they tend to swim in circles if there is a sudden spike in temperature, something I observed. They also have a very interesting ability called phototaxy, or phototactic ability, meaning they swim towards or away from the light, depending if it's positive or negative. So, for example, if we know that they all swim towards the light when everything is okay, if most of them don't swim towards the light, then this is a sign of a pollution or any other stressor in the environment. Of course, mortality, if they all die at once, that's also a sign of stress. And last but not least, we have hemoglobin accumulation. So in low oxygen environments, after a longer period of time, they tend to produce more hemoglobin because it's more efficient and it's carrying oxygen throughout the body. And because it's red in coloration, they turn from clear to this red color that could be quite interesting to look at with a camera. And last but not least, we have the microinvertebrates community structure. They could be benthic or planktonic. Benthic means they live on top of the sediment or within the sediment, and planktonic meaning they swim in the water column. So we know that certain trophy levels in lakes present different community structures. So certain species dominate, or certain orders dominate, or certain species are present or absent. So with increasing trophy, or if any changes in pH or temperature occurs, this structure will change. So we'll be able to observe different species in different orders dominating. But here, the difficulty of perception is quite high because we're not even looking at what species anymore, or even one order. We're looking at a whole community structure. So this could be quite challenging to detect automatically. There are some ideas that are being developed, for example, glass tubes, and that the organisms could crawl through or swim through, and then they could be automatically sort of identified with a camera. But again, this needs to be developed further. Now a little bit about the power source that we're planning on using, about the microbial fuel cells, or MFCs for short. This is what the MFC typically looks like. And what it does is it extracts, it uses natural processes that happen in the mud and extracts energy from bacteria breathing, basically. This is more or less how it looks. It's a very simple schematic just to show how it works. It's consisting of 2 chambers. The important part is that one of them is anoxic in the sediment, so there is no oxygen present, and the other is exposed to oxygen, so exposed to the water. And what it does is basically bacteria is breathing anaerobically in the sediment, and the electricity is extracted through that process. So this is just the basic gist. So if they're so amazing, why are they not used wildly everywhere? Well, the main limitation of an MFC is its low current. So it's usually not enough voltage to power anything of significance. And we also know that they don't scale well at all. So larger MFCs don't produce more power, so they have a limit. And we also know that connecting multiple MFCs to each other will cause something called voltage reversal. So the whole system will get shut down. But nevertheless, they've been used mostly in the lab in the past. For example, Walter Group managed to use MFCs for powering microcontroller. I believe it was a Game Boy for about 100 hours. So that was quite successful. Later on in the field, it was used by Bristol Robotics Laboratory, where there was a famous case. They used MFCs to power bathroom cubicles using a urinal during some sort of music festival. And also they were used in the aquatic field by the project Subculturon, which was a project that occurred before Robocenosis. And what they did is they used 6 stacked MFCs. So the MFCs were still producing electricity sort of separately, but they were all harvested by one supercapacitor, so-called. And this energy was already enough to power a robot, which is something which we'll also be attempting with Robocenosis. Quickly about biodegradability, we'll be using various biodegradable materials like wood, paper, for example, rocks as weights, cotton, and many, many others. Here is a simple example of how we'll be using those materials. For now, we're using whatever we need to use as a sort of proof of concept, and mostly using wood and paper. And later on, we will sort of translate the whole machine to be biodegradable. A little bit about where we work. We'll be working in 3 Austrian lakes, 3 beautiful Austrian lakes, and they're all protected areas. So it's really great that we have allowance to work there. First up, we have Milchstädter See. It's located in Carinthia. It's glacial, meaning a glacier took part in forming it in one way or another. It's oligotrophic, meaning the nutrient content is very low, so the water is very clear and of actually drinking quality. And it's also metamictic, meaning that there are layers of water that never undergo mixing. Next, we have Hallstätter See, which is located in Upper Austria. It's also glacial, but it's oligomesotrophic, meaning the nutrient content is still pretty low, but it's a little bit higher than just oligotrophic. And that is mostly influenced by salt mining that happened in the past. And lastly, we have Neusiedler See, which is a completely different habitat to the other 2. It is located in Burgenland, in, again, Austria. It's eutrophic, meaning the nutrient content is very high. And it's also extremely shallow. So the water depth never exceeds 1.8 meters. So you could technically just walk through it. And it's also endorheic, meaning there is really no major outflow. So the water level is regulated mostly by precipitation. This is a very interesting habitat to work in because, for example, here in the open lake, the water visibility is 0 or close to 0. So using things like cameras will be challenging, if not impossible, really. Here is the setup that enabled us some early experiments that we carried out. Here is a watertight tube with a Raspberry Pi and a Raspberry Pi camera module within. Again, this is what Raspberry Pi is. We also have some various sensors provided with the setup, like conductivity, temperature, and pressure. These are mostly for sort of proof of concept, so we need to be able to test if the life forms are behaving the way we think they're behaving. There is an on and off switch and pressure relief valve. So this is basically how the tube looks during the experiment with all the electronics inside. What it does, it's constantly looking at the muscle. We used a simple red marker to later on track the movement of the valve. As you can see, this muscle is pretty big. So it's quite simple to see it with the camera. Again, here is a simple red marker. So instead of tracking the whole muscle or the whole even valve, we're only focusing on this one center. And this will make the tracking of the movement of the valve much easier or much more precise. Here is how the last process image looks. There might be some wasted space all the way around the muscle, but in the future we'll be planning on using multiple muscles and again multiple life forms, so maybe there's something in the background to be seen as well. Here is the same setup, but using zebra muscle. As you can see, it's much smaller. This is the black dot right here. And here we used something very similar. So again, the simple red marker, but we added a benchmark. So again, this is what the camera sees. This is the red benchmark. This is the red marker. The zebra muscle is behind it. And instead of tracking the movement of the marker, we're constantly tracking the difference between those two benchmarks. And again, there is a lot of space in the background, but this will be dealt with later on. Here is a very similar setup, but using Daphnia. So the small crustaceans that I showed earlier. There are two setups. Here is again using the plastic tube, and here is a more of a crude version with a shrimp cage. So the idea is that we put the whole setup in the water. You pour the sample with animals through this hole, you plug it, and the camera is continuously observing their swimming behavior and swimming patterns, and they can be analyzed later on. And again, we're using rocks as weights, for example, here. Very quickly about the release mechanism. So the mechanism that will make the non-biodegradable part float to the surface. Again, it will be made of biodegradable materials. So for example, rock as a weight, rock that is taken from the lake. So we're not really introducing any external materials, or it's very limited. We're using, again, wood and paper, multiple layers of paper. So the only feature of this release mechanism is to just return the device within a certain time frame. So it could be a couple hours, it could be a couple months. For now, we've been testing hours. So multiple layers of paper, the more layers, the longer it takes for them to disintegrate. And once they do, a sort of a pin is released, and the floating device will float upwards after the weight has been released. So just to sum up, what we're working on here with the Robocenosis project, we are attempting a remote real-time continuous monitoring. We will be using a concept called life form in the loop. So using life forms instead of classical sensors, which will sort of give us an ability to hack into the ecosystem. We will be using a low power autonomous setup and energy harvesting for this. And the part that remains in the environment will be completely biodegradable. So the impact of the research will be pretty much non-existent on the environment. Thank you so much for listening, and I'm happy to take any questions.
Yeah, thank you, Victoria. I had problems finding my mute button. Very, very nice talk. Reminded me a little bit on Leonardo. I guess he was also like crafting from stone and wood and these materials. So somehow also a little bit back to the roots in robotics. Do we have questions? Unfortunately, we only have time for one question.
Only appreciation comments in the chat.
Yeah, I'm looking, but I'm currently fighting with my Zoom windows a little bit. Yeah, but very, very simple question, Victoria. Is this something that can scale up? So could you do something like a robot swarm, for example? Because I guess for environmental monitoring, probably more than one device would be needed. And is it then just throwing in multiple, or would you think that there could be any sort of interaction also possible by following this paradigm?
I think it's definitely possible, especially that we're, at least I'm focusing on making it modular. So using a lot of different life forms. And if it turns out that maybe it's not practical for one robot to have 5 different life forms on it, maybe making it a swarm could be more logical, I guess.
Yeah, okay, thank you. We have one question in the chat. The question is whether you think it would be possible to make biodegradable materials also in the fuel cells. So let them be biodegradable themselves.
I think so. I hope so, at least. I'm not an expert on the MFCs, unfortunately. This is what our brilliant engineers are working on. I hope it's doable. I also heard that there is an idea of using algae as well, instead of bacteria. That's another idea.
But I think there are MFCs, paper MFCs in development. I think I have seen some of them. If these papers are then biodegradable, I don't know, I have to say.
Okay.
Okay, thank you, Victoria. Unfortunately, we run out of